Hello everyone, my name is Donya Vaez and I'm a senior in the MSCC program here at Dunbar. I had the pleasure of working under the mentorship of Dr. Frank Gillum in the UK Department of Neuroscience. My project title is a transcriptome analysis of dual pathology in epilepsy. An overview of what I'll be talking about is the background information, question hypothesis, methodology, data, conclusions, future research, acknowledgements, references, and I'll finally open it up to any questions that you would like to ask via email. To get started, epilepsy is defined as 2 or more unprovoked seizures. This disorder affects about 1.2% of the US population, which is about 4 million people. Normal activities such as driving, swimming, and cooking all have to be restricted in order to ensure the patient's safety. Even though there's a lot of medications available to epileptic patients, about 40% of these patients cannot be controlled with the current medications, and they can become quite expensive and hard to afford by families. Some patients qualify to be focal epilepsy surgery candidates in order to help reduce the frequency of their seizures, and their tissue is used for research like it was for my project. Within focal seizures, temporal lobe epilepsy is the most common epileptic disorder, with more than 50% of cases being associated with it, starting in the hippocampus. Hippocampal sclerosis is the most common pathology associated with temporal lobe epilepsy, or TLE, with the loss of neurons and granule cell dispersion. MMCD is another pathology associated with hippocampal sclerosis in the temporal lobe neocortex. This is called malformation of cortical development, but it's also abbreviated, which you will see a lot in my presentation, to MMCD. Dual pathology is cases that involve both MMCD and hippocampal sclerosis, so they will have both of the pathologies. And these 3 groups will be mostly focused on in the data collecting and RNA-seq of my project. In this dual pathology patient's MRI, you can see that the brain on the left and the difference in the white matter of the right temporal lobe brain and the difference of the left one, that there is definitely a difference between them, meaning that the temporal lobe MMCD is prevalent right here. And on the brain on the right, you can see the shrunken hippocampus on the right compared to the larger one on the left. And these MRIs are like you're looking directly into them, so that's why it's backwards, right, and left. For our project, some specific terms to note are somatic mutation, which I have a diagram of on the next page, but they are genetic alterations during cellular differentiation. The MMCD pathway associated with the SLC35A2 gene, also known as the glycosylation gene. 20 to 40% of patients with MMCD pathology were also associated with the SLC35A2 gene, which made me interested in looking in the different transcriptomes of these patients. Somatic mutations guide the importance of glycosylation defects. So this means that's why I looked for the SLC35A2 mutation and quantified the abnormalities with differential RNA libraries and biochemical pathways. The differential RNA found abnormalities encoded by the SLC35A2 gene, or abnormalities in the transcriptome pathway related to the SLC35A2 gene. These abnormalities cause biochemical glycosylation defects in the Golgi apparatus and intracellular functions to be affected, which is the main focus of the project. So this figure was made by me in order to primarily show the somatic mutations are made after ectodermal differentiation may cause brain pathology. So in this MRI right here, you can see the drift pattern of a patient's MRI that had MMCD. And you can see in the pathology on this column in the L3 where this arrow is pointed, if there are 8 or more neurons stacked on top of each other, that is defined as MMCD. So here is a visual of the MRI and pathology that are compared in order to identify a patient with MMCD. So my investigative question is, do the changes in the transcriptome concur with the SLC35A2 mutation in the hippocampal sclerosis, MMCD, and dual pathology cases? And my hypothesis was that variability of the RNA expression for the specific isoforms, especially the ENST one that we have identified, are associated with the SLC35A2 gene, and they will identify abnormalities in each of the pathologies, which we have 3: MMCD, dual pathology, and hippocampal sclerosis. So for my methods, the RNA-seq of each subset of samples were completed and stored in their own library through this software program used for RNA-seq that's pretty common, called the Reactome. So in the Reactome, the RNA libraries for each of these subset samples, which were those 3, were compared to one another, determining the abnormalities of the biochemical pathways, basically. More specifically, the MMCD, or mild malformation cortical development samples, were compared to the normal cortex and dual pathology sample, where the map of the entire library of these intercellular pathways were compared through upregulation and downregulations of the genes. Specifically the SLC35A2 gene, but other genes such as mTOR, which is not related, were compared, including FCD, which is focal cortical dysplasia, which we'll get into. Sorry about that. This data table shows the different groups divided in columns: MMCD, TLE, and dual pathology. MMCD had a mean age of 42.9 years old, with 30% of the patients being male and 69% of these patients being female. Temporal lobe epilepsy had a mean age of 38 years old, with 56% of these patients being male and 44% of them being female, with the dual pathology cases, including both TLE and MMCD. Even though we only had 4 patients, which is a limited study sample, we had about an average of 38.3 years, with 2 of 4 of these patients being males and 2 of these being females, splitting the percentages 50-50. But in conclusion, there were no statistically significant age and sex differences between these groups. Sorry about that. This graph shows the different groups of variability with the specific isoform ENST0000376521 6 isoform that is specific to the SLC35A2 mutation. And this is actually just 1 out of 18 of the isoforms associated with this gene. And you can see in the blue, we have temporal lobe epilepsy. The orange is our focal cortical dysplasia, with our control of being the green, which is just 2 patients, and they are obviously at 0, which is what we expected in the variability. So the variability was measured in copies per million of the specific isoform of our SLC35A2 gene. So as you can see, about 14 of the 17 cases were upregulated, increased expression, with about 1 out of 5 of the cases of focal cortical dysplasia and the controls being upregulated, which is just this 1 compared to all of these 5, 2 here, 2 here, and 1 there. So that's 5 over there, with only 1 of them being upregulated, meaning that these 14 out of these 17, with the 3 of them being down here, were upregulated compared to only 1, despite the lower sample size of focal cortical dysplasia patients, was upregulated. This means that there's definitely a specific change in the transcriptome between temporal lobe epilepsy and focal cortical dysplasia patients, because the visibility and visually you can definitely tell that the temporal lobe epilepsy patients had definitely a higher variability in the specific isoform of our gene. So I used the Pearson's chi-squared test to determine the p-value between the upregulated and downregulated samples of temporal lobe epilepsy and focal cortical dysplasia or normal. So the plus expression, as you can see over here, means upregulated, and the negative expression means downregulated. About 14 of them were upregulated, as I told you, and 3 of them were downregulated, with 1 of them being upregulated for FCD and normal. These groups were just combined, with 4 of them having downregulation or they stayed at 0. So basically, these were calculated, and the chi-square statistic was 6.9, giving us a p-value of 0.009, which is statistically significant, with p being less than 0.05. So this means that our data for the TLE and FCD samples of their variability of the specific isoform was statistically significant. So basically, the hypothesis of variability between the 3 different groups of temporal lobe epilepsy, mild malformation of cortical development, and dual pathology was supported due to the different transcriptome abnormalities between TLE and FCD, as you can see in the graph, where about 14 out of 17 were upregulated for temporal lobe epilepsy, but only 1 was upregulated for focal cortical dysplasia. So the RNA-seq showed that it was supported that the SLC35A2 mutation was presented in the temporal lobe epilepsy samples in great variabilities. Upregulation of this ENST0000376521-6 isoform of the SLC35A2 gene was proved with the abnormalities of the temporal lobe epilepsy and focal cortical dysplasia, and this basically proves that this specific isoform changes indicate abnormal regulation of the galactose translocated in the Golgi apparatus, which is what we were looking for to set it apart from different neurotransmitter and ion channel pathways, biochemical pathways. Sorry about that. So for future research, completing these causes of MMCD gives us a more complete understanding of what actually it means to develop this pathology. And this will identify different pathology than the more conventional of ion channels and neurotransmitters, such as glutamate, that are the targets of what epileptic medication is targeted to help mitigate seizures by. So this way we can identify new therapies and medications despite the ion channels and neurotransmitters, looking more specifically into biochemical pathways associated with the MMCD and looking specifically at what isoforms need to be targeted and such. So also this can be used in other epileptic disorders, not only temporal lobe epilepsy, but also specific ones such as Sturge-Weber and many more. Here are my references. And I would like to first and foremost thank my mentor, Dr. Frank Gillum, in the Department of Neuroscience. He has taken me underneath his wing for the last couple of years and has really showed me the amazing world of neuroscience, and I hope to pursue a career in it in my future. Thank you to the University of Kentucky, the Department of Neuroscience, and the Kentucky Young Research Program to allow me to do college-level research in high school. Thank you to Ms. Karen Young, all the other teachers in MSTC and the MSTC program as a whole for supporting me and giving me this opportunity. Thank you to my family and friends for supporting me all through this journey and never letting me give up. If you guys have any questions, feel free to email me at donyabaez@gmail.com, and I will try to get back to you as soon as possible. Thank you guys, and I hope you guys enjoy. Have a great day.